Hello, our today lesson is about nucleic acids. We will know about several concepts that are nucleotides, the difference between DNA and RNA, nitrogenous bases, and the DNA structure, and types of RNA, Watson and Crick, and DNA organization. First, the nucleotide is composed of three main parts. The phosphate group here in yellow, the pentose sugar in purple, and the nitrogenous base that is represented here by a blue color. These are the three main parts of a nucleotide. So DNA or RNA, both are composed of monomers that are called nucleotides. These nucleotides, again, what they have a phosphate group here, they have a sugar, and a nitrogenous base. This nitrogenous base could be one of five types of nitrogenous bases, either adenine, cytosine, guanine, thymine, or uracil. And we have a pentose sugar here. That means what the meaning of pentose? It means that it's composed of 5 carbon. Its structure is composed of 5 carbon and a phosphate group. Let's illustrate the difference between DNA and RNA. For DNA, it's a deoxyribose sugar. What the meaning of deoxyribose? In case of ribose for RNA, it has OH group in the second carbon and the ribose sugar. Whereas in the DNA, it's deoxy, which means it's removed of oxygen. It's devoid of oxygen. The oxygen is removed here. It's only H. Okay? So in DNA, it's a deoxyribose sugar, while for RNA, it's a ribose sugar. For nitrogenous base, we have thymine for DNA, and it can present in DNA only, but not present in RNA. For RNA, there is uracil that's present in RNA only, but not in DNA. DNA is double-stranded and has the name of double helix, while RNA is a single-stranded. What is the basic function of DNA? It's the master copy of genetic instruction that full copy of all instructions inside the cell is present in DNA, while RNA is only a temporary copy of a specific instructions that's used as template to generate or to synthesize proteins. Nucleic acid composition, as we said before, we have 3 main parts. Can you see them? Yes, that's right. The first one here is phosphate group, and we have this sugar, and we have nitrogenous base. And what's the most important to look for as sugar? It's ribose sugar. It's not devoid of oxygen. It's OH, so it's not deoxy. It's ribose sugar. And for nitrogenous base, what's common? A, C, and G are common for both DNA and RNA. But for RNA, there will be uracil that won't be available for DNA. And for DNA, you will have the deoxyribose sugar and the phosphate group and nitrogenous base A, C, G, and T, which stands for thymine, which is present only for DNA but not for RNA. Polynucleotide formation. The polynucleotides formation nucleotides are linked through single polynucleotide strands via condensation reaction, and water is produced in this condensation reaction. It's a condensation reaction between the OH group on the carbon number 3 on sugar, and the OH of phosphate to form phosphodiester bond that's represented by red line here and there. The 5' phosphate group of one nucleotide attached to the sugar of another nucleotide on the 3' hydroxyl group, and the nucleotide strands will run in 5' to 3' direction. And the bond that is formed between the two nucleotides is called the covalent phosphodiester bond. These are the nitrogenous bases that are present in DNA and RNA. Adenine and guanine are double-ringed purines. Cytosine, thymine, and uracil are single-ringed pyrimidines. The DNA molecules from double helix structure forms a ladder with the sugar backbone that are connected by bases. And the bases, a ladder here, the backbone here from phosphate sugar backbone. That are the two sticks of the ladder, two main sticks of the ladder. And the steps of the ladder represents the nitrogenous bases in perpendicular on the main sticks. The nitrogenous bases from each of the two strands form a complementary pairing connected by hydrogen bonding. Cytosine and guanine forms 3 hydrogen bonds, and the pairs of adenine with thymine forms a pairs of 2 hydrogen bonds. Forms pairs of 2 hydrogen bonds. Any mismatching in pairing will destabilize the DNA ladder. In order for two sets of nitrogenous bases to pair, the two strands must run in anti-parallel structure, one from 5' to 3', and the other from 3' to 5'. And there is twisting in order to adopt the most stable energy configuration, and DNA forms stable double helix arrangement roughly from 10 to 15 base pairs per rotation.